It's my pleasure to welcome the next speaker, Rosanna Olsen. So thanks a lot for giving this presentation today. So after a PhD in psychology at Stanford University and a postdoctoral training at Rotman Research Institute in Toronto, you are now an assistant professor in the Department of Psychology at the University of Toronto. You are very involved in the hippocampal subfield group, and you also work on the contribution of the anterior and mediodorsal thalamus to memory. So thanks again for giving this presentation, and you can share your screen whenever you're ready.
Okay, I don't think I've been, oh, I have been given permission. Thank you. I see it.
And we can hear you perfectly.
Oh, great. Okay, so let me just go into presentation. Okay, and can you see my screen okay? Yep. I didn't see, okay, good, thank you, thumbs up. Perfect, so I'm gonna try to do 2 talks in one today in my 15 minutes. I'm going to speak about the topic that was on the presentation, the first slide of my presentation, which is the roles of the anterior and medial dorsal thalamus in memory. And I'm also going to mention some of my involvement with the hippocampal subfield group, because I think that some of the issues that we've tackled as a group are very relevant to many of the people on the call today. So I first started studying the brain and started studying memory, very much focused on the hippocampus, like many of us in the field. And coincidentally, while I was at Stanford, I worked with Gary Glover and my supervisor, Anthony Wagner, as well as other members of the lab, to develop methods for segmenting and analyzing different subregions of the hippocampus and adjacent medial temporal lobe cortex, as shown here. But more recently, my interests have traveled to another part of the brain, primarily to the thalamus and the extended hippocampal system. And as I said, a lot of the issues that are germane to hippocampal subfield segmentation and visualization of the substructures seem to be very relevant to the challenges that are faced by those who would like to look at the thalamus. So I hope to offer some wisdom there. So just to talk a little bit about how I got interested in this area of research, I had the opportunity to collaborate with Shana Rosenbaum, who's a neuropsychologist at York University, who studied several different cases of people with amnesia, both acquired and developmental amnesia. And in particular, I started collaborating with Shana to study this particular case HC, who is an interesting case because she has bilateral hippocampal volume loss, as shown here, as characterized in this high resolution imaging of the medial temporal lobe. But she's also interesting for another reason, and that is that Shana and Fucheng Gao, who is a neuroradiologist at Sunnybrook Hospital working with Sandy Black, has shown that in addition to that volume loss, that bilateral volume loss in the hippocampus, she actually also has significant abnormalities to the extended papas circuit and extended hippocampal system. So for example, she has abnormalities to her fornix, to her mammothalamic tract. She has reduced volume in her anterior thalamus, and she also has, at least in using standard T1 weighted imaging, we cannot visualize her mammillary bodies, which has led Shana and others to speculate that the etiology of her developmental amnesia might be something that happened in utero, perhaps, although that's not completely agreed upon. However, I will note that others, including Varga Kadem in England, who have also done very careful studies of people like HC and other individuals with developmental amnesia, have also noted abnormalities to the anterior thalamus. I've also had the opportunity to work with another case of developmental amnesia, and he's known in the literature described as case NC. And case NC, actually both case NC and HC are young adults, and they both acquired their amnesia, you know, in development, which is why they're called developmental amnesics. But case NC is very interesting because, like one of the flash talks described earlier, his amnesia actually developed not at all from damage or volume loss to the medial temporal lobes, but instead due to an infarct that occurred when he was 9 days old. And he suffered a lesion in his medial dorsal nucleus of the thalamus here on the right, and the radiologist and neurologist in the room can probably see there's also affecting his left medial dorsal nucleus of the thalamus. Now, unfortunately, we don't have this quantified using segmentation methods, either manual or automated segmentations, but this was the assessment by the neuroradiologist. So I started getting interested in studying these two regions using fMRI, which is my primary approach for studying the cognitive neuroscience of memory, because I realized that these two cases were very, they were very transformative in my thinking about how memory is supported by the brain, and made me realize I had to focus outside of the medial temporal lobe. And I will note that HC and NC, like I said, they're around the same age, and they actually show remarkably similar neuropsychological profiles in that they have this very selective episodic and autobiographical memory impairment, whereas in the rest of their cognition is fine. So I told you that these two patients, case HC and case NC, have different volume losses in the anterior nucleus of the thalamus and the medial dorsal nucleus of the thalamus. And I won't go into too much detail about what we think these different nuclei are doing, because many of the other talks in this series have already covered that. But I will say that our thinking on this at the time, at least, was very much focused on some of the writings of Aggleton and colleagues, pointing to this potential role and a potential dissociation between the ATN and the MDN in associative memory and familiarity. And that is driven by differential inputs from the medial temporal lobe substructures into the MDN and the ATN. So my student Kirk Geyer and I conducted this fMRI study where we collected whole brain imaging during a memory task in which healthy young adults were being scanned while they encoded face and scene pairs. And then we also scanned them during the retrieval phase when they were shown, which I'll explain in a moment. And we collected, again, whole brain 2 millimeter isotropic imaging. And we focused in on these two nuclei, the anterior nucleus of the thalamus and the medial dorsal nucleus of the thalamus, using the Morel Atlas of Krauth. And first, I just want to, first I'll describe the task a little bit more. So it's an associative memory task in that they had to form associations between the faces and the scenes. And then it was a little bit different from a standard recognition memory task in which we didn't just show, the images and ask whether they were old or new, but we actually cued participants with the scene that was previously paired with that face. So this is eliciting, we asked participants to try to remember the face that they saw with that scene previously during the study phase. And then we had a delay phase in which participants had to remember and recall that face that was previously associated with this scene. So during this phase, they're just looking at a blank screen and they were supposed to be remembering this face. Then the scene appeared again, the same scene that they just were cued with, and then 3 faces that were previously studied were shown, and they had to decide which of those 3 faces was the match to that scene. So I'm just going to show you the entire brain, not masked first to the ATN and MDN, just to show you sort of what's going on during different phases of the task, because not many people in the literature have really focused and zoomed in on the thalamus. So I just want to show you in the context of all the other areas that are active in the brain. So, so during the study phase, of course, the hippocampus was active in forming that memory association. Parahippocampal gyrus is on fire. And then instead in the thalamus, what we saw is actually a deactivation in the anterior nucleus of the thalamus, at least in that region, to the best of our abilities to localize. And during the test phase, that's when we saw regions, specifically actually during the delay phase of the recall phase of the task, was when we saw the thalamus showing greater activity compared to baseline. So specifically during the task in which they were trying to remember the person's face that was associated with that scene, we saw robust activity in the medial dorsal nucleus of the thalamus and the anterior nucleus of the thalamus. And then again, during the memory decision phase, we saw still elevated activity in the medial dorsal nucleus of the thalamus. So this is just main effect activity, not differentiating between correct and incorrect trials, and that's what I'm going to show you now. So I'm going to show you time courses during that retrieval phase of what the activity in the ATN and the MDN looked like during that time, separated for correct and incorrect trials, similar to Dr. Sweeney Reed this morning. So what we saw here is that in the anterior nucleus of the thalamus, we saw greater activity for correct trials during this delay phase in which participants were trying to remember the face. And that was significantly greater for correct trials compared to incorrect trials. The activity was, as I said, above baseline in the MDN during this time, and especially it was the greatest during this memory decision phase in which participants were trying to differentiate between the three familiar faces. However, the signal didn't differ across correct and incorrect trials, suggesting that this region of the brain was not differentially active or involved in that memory process in the same way that the ATN is during the delay phase. So just to recap this study, what we saw was that the anterior thalamus was active during the memory recall phase, and that activity was associated with memory accuracy. And I mentioned to you that the anterior thalamus was deactivated during encoding in that it showed lower bold activity compared to baseline. And in fact, what we saw is the lower the bold activity, the higher the probability that that trial was correct. So more deactivation at encoding was actually associated with subsequent memory. And the medial dorsal nucleus of the thalamus was activity was increased during memory retrieval, especially during that memory decision phase, but it was not associated with memory accuracy. So we're currently in collaboration with Kirk, we're currently conducting some follow-up analyses now that we have sort of, and Lola mentioned this earlier in her flash talk, we're starting to think a little bit more carefully about the role of the medial dorsal nucleus and its connectivity with the prefrontal cortex and what role it might be playing in cognition. And we're looking specifically in patients with MD damage to see if there are alterations in intra-PFC functional connectivity. So now I'm going to switch gears and I'm going to talk about harmonization efforts from the hippocampal subfield group. And for those of you who aren't familiar with the group, I'll just briefly explain who we are. So it's a group of researchers it's an international working group, and we have been working together for quite some time. This is the first paper that we published together in 2015. And this paper was led by Paul Yushkevich, who's at Penn. And the reason why we published this paper is because, as you all know, there have been many, many studies of hippocampal involvement in memory and in aging and Alzheimer's disease, and many, many different labs around the world have been studying the hippocampus using, for example, a high-resolution MRI, like that example I showed you in the very first slide. However, we recognized that there was a serious problem in the field because even back in 2015 and now 7 years later, you can imagine the problem is even worse, that there were very much different approaches to the way we were delineating the different subregions of the medial temporal lobe. And so what we did is the group of us, the 21 different labs that contributed to this figure, we took the same brain and we all applied the protocol, the manual segmentation protocol or the automated segmentation protocol that we use in our lab. We applied that to the same brain to show and illustrate the differences, sometimes very dramatic differences in both the number of regions that are segmented and where the boundaries are placed in between the different regions in the medial temporal lobe. So for example, this is the segmentation protocol that I use in my lab. And you can see there's some similarities between other labs that may have trained with the same mentors, for example. But there's vast differences between some of those protocols and some of the other protocols that look at very much, in fact, both a different number of regions and also placing those boundaries in different regions. And so I was not aware of this figure in this paper until earlier this week, but it was very much reminded me of this 2015 paper that that Paul Yushkevich led, where in this paper by Jürgen May and Nashchinek, they have showed that different neuroanatomists have segmented the thalamus subregions in very different ways. And Jürgen has posited some of the different reasons that you might expect that this could occur. So those reasons we think are also obviously affecting us in the hippocampal world. So one potential reason that differences exist across the protocols is that The neuroanatomists who have developed the protocols have used different individuals, for example. So we saw in Eric Middleton's talk, Middlebrook's talk, that there's individual differences across even healthy individuals. So if one atlas was developed on one person and another atlas was developed on another person, could that have resulted in the differences between some of these atlases? That's one reason. Another reason is that different methods, of course, are used. So different types of stains are used across neuroanatomy labs. Some neuroanatomy stains are better at picking up cell bodies, for example, versus myelin versus other properties of the tissue. And so that has also led to differences. Another difference is, I mentioned this a few days ago, the plane in which the slice is made could affect how you visualize these different regions. So for example, in the hippocampus, if you're in the body of the hippocampus, it really doesn't matter how you slice it. Sort of the orientation of all the different subfields is sort of the same. But once you get to the head or the tail, which are curved structures, the plane in which you're sampling is very, very important and will very much determine how you can visualize and where the different positions of the subfields are in relationship to one another. The other difference is sort of just schools of thought. So it could be just that different schools are cueing onto different aspects or different properties that are, and that sort of goes along with the methods as well, could be driving these differences. Another thing is probably that different samples might have gotten damaged or were based on, for example, clinical populations. So they showed this paper shows one example where Parkinson's patients will obviously look a little bit different than a healthy adult. So you have to keep all of this in mind when thinking about the atlas that you're choosing to develop your protocol. So does this matter in terms of how we study the brain? Well, we obviously think it does. And one of the reasons that Robin DeFlores published this paper in Neuroscience in 2016, which is a very eye-opening paper because it goes through, and this actually might converge with Michael Hornberger's question for Brian about, why didn't you see any differences in the hippocampus with age? And this figure is demonstrating, it's a visual depiction of a table that's in Robin DeFlores's paper that shows that across all of these different studies, yes, many of these studies did show differences due to age in subregions of the hippocampus, but look how different the findings were across the different studies. And in fact, there's one study that didn't show a difference in hippocampal volume with age. But what's shown here is the yellow is studies that use T2 weighted imaging, and the red is studies that use T1 weighted imaging. And what Laura has done is she's color coded the different subfields that have shown differences across different studies, and you can see that they're very different. One potential explanation for this is not, of course, the protocol heterogeneity, but it could also include the samples that are chosen for the study. So that's, of course, important to keep in mind, but we don't really know if that's the case because we can't really compare. It's like comparing apples and oranges if you have different protocols. So what has the harmonization group done? What has our harmonization effort looked like so far? Well, we've teamed up with a group of neuroanatomists to collect labeled histology to try to understand where some of those differences across the atlases are coming from. And so what we've done is we've collected histology that's labeled by different neuroanatomists from different schools so that we can help us understand where the differences in the atlases are coming from. Then we've done this so far. This is sort of just a little schematic to show you how far we've gotten. So we started in the hippocampal body. We've collected that histology in conjunction with neuroanatomists. We've developed some rules for subfield boundaries on high resolution T2 MRI. We've done an initial feasibility check, at least for the body, to make sure that the rules that we're developing make sense. And now we're using a Delphi method to collect feedback from the community to make sure that it's suitable for their needs. And then finally, we'll do a formal reliability analysis. And this process is also occurring in the hippocampal head and to some extent in the hippocampal tail. Now, the hippocampal tail is a very complicated anatomical structure, and it's very curved in some individuals. And some hippocampal segmentation protocols, including FreeSurfer, have decided not to divide the different subfields in the tail due to the fact that it is so complicated. And good consensus doesn't really exist in terms of where the subfield boundaries lie. And so that's actually the approach that we've taken here as well. So who are the neuroanatomists that have been involved? Ricardo Insausti, Olga Kito, who works closely with Katrin Amons in Germany, Sonling Ding, and Jean Augustinac are the neuroanatomists who have very kindly been involved in this effort. And so for for the development of the protocol, we've had at least 3 neuroanatomists label the different regions of the hippocampus. And so this will at least take out the individual differences variability that I mentioned before, because it's the same sample being labeled by 3 different neuroanatomists. But you can see even here, even using the same stains and the same subject, you can see that the number of ROIs that are, the number of regions of interest or the number of subfields that are divided are different among the neuroanatomists, because they don't all agree on the terminology and number of ROIs that exist in the hippocampus. And the boundaries also differ somewhat from group to group. And we've been very fortunate to work very closely with Sonling Ding, with all, in fact, with all 4 of the neuroanatomists. They have come to our working group meetings to help us learn more about their techniques and also to help us guide the development of our protocol. So how will we know that the harmonization effort worked? This will kind of harken back to Manoj's talk on the first day. So we can use different methods to try to understand if our harmonized protocol is indeed working better than the existing protocols that we've all been using on our labs. So one type of validation we call bronze standard, which is, for example, we can apply our new harmonized protocol to people with MCI and Alzheimer's disease who have measures of their neurofibrillary tau pathology, like in ADNI, to see if our protocol can predict further neurodegeneration caused by the accumulation of tau pathology. So that's one way that we'll test this. And then another way is to look at co-registered labeled histology that is co-registered to ex vivo MRI. And we're very lucky and fortunate that such data sets exist and are publicly available. So Paul Yushkevich, our collaborator, and this paper was one of the first papers that came out on this using this technique by Adler and colleagues in PNAS in 2008. So what they've done is they've collected a number of post-mortem brains where they have labeled histology co-registered to their ex vivo MRI, very high resolution ex vivo MRI. And this is actually a publicly available data set, which is great. And hopefully something similar could be developed for the thalamus. So just my final musings on the, since I am I took the liberty, since I am the last speaker of the series, and thank you so much for everybody for staying here to watch the final talk. So first of all, is harmonization possible either in the thalamus or in the hippocampus? I would say yes, but it's very difficult. And it's a process that has taken quite some time from our international working group. And I imagine it could be very difficult to achieve this kind of thing in the thalamus as well, but I think it's a worthwhile endeavor. I also wanted to point out that we're all sort of searching for, well, what's the best imaging protocol? What's the absolute best analysis method? And I just wanted to point out that there might not be a one-size-fits-all imaging and analysis solution, and that when you're planning your studies, you should really try to think about the question and the population that you're analyzing, and that your protocol and methods should be tailored to the question at hand. And obviously, if you're studying Wernicke's amnesia versus schizophrenia versus another disorder, the type of imaging imaging protocol and analysis that you might choose might differ across disease or population. I also wanted to bring up this question, which or this topic, which hasn't really been broached that much except for by Eric earlier today, in that there's currently a little bit of a debate in the neuroscience literature about sort of, well, what's the most meaningful delineation of these regions, such as the hippocampus and the thalamus? Is it really going to be based on the parcellation schemes that we've all come up with based on cytoarchitecture, or are there other more meaningful functionally based delineations that might be important to consider? And gradient mapping methods have recently been applied to areas like the medial temporal lobe, maybe in the thalamus, but I'm not sure, but also obviously to the neocortex as well, showing that maybe there's not stark differences between one region and the next, and depending on the region of the brain that you're looking at. And again, I'm not sure this applies quite as much to the thalamus, but in the cortex, at least, sometimes there could be some more smooth gradients between these different regions. And so this is just a paper by Sarah Gennon that addresses this issue. And I'm happy to take any questions and start the roundtable. Thanks.
Thanks a lot, Rosanna. Okay, thank you.